Okay, so today we're going to be looking at DNA structure and replication and protein synthesis. So DNA stands for deoxyribonucleic acid. It has a double helix structure. This three-dimensional structure was first determined in 1953 by James Watson and Francis Crick. They would not have been able to discover this structure if it hadn't been for the help of Rosalind Franklin and her X-ray crystallographic image, which I will show you in just a minute. So Watson and Crick, of course, became very famous and shared the Nobel Prize in 1962 for the discovery of the structure of DNA. Rosalind Franklin, however, did not share in it as she had died in 1958 of ovarian cancer. The Nobel Prize is never awarded posthumously, so she did not get the Nobel Prize, which is a real shame because they would not have been able to get it without her help. There was a rush at the time. Lots of people in lots of different countries were working on the structure of DNA. The French were very close to winning the race, but Watson and Crick did it because of the image by Franklin. And this is a picture of Rosalind Franklin here and her X-ray crystallographic image. And you can see on the X on this image that that was the information that they needed to be able to determine that it was a double helix structure. And this is Francis and Crick in front of a model of the DNA molecule. So one of the really cool things about the DNA double helix is that this structure, this double helix structure, makes it very easy for DNA to replicate itself. Not because it's a double helix necessarily, but because the bases are held together with hydrogen bonds. And as we've talked about before, hydrogen bonds are a weak bond. They're very important biologically, but they're a weak bond. So these bonds could easily be broken, and as they're broken, the two halves of the DNA can unwind and allow for the duplication of it. So this is a really great example of form versus function. And of course, because DNA has to replicate itself whenever cells divide, as we've talked about with mitosis and meiosis already, this is an excellent way for them to do that. And also DNA gives the molecular explanation for genetic inheritance, which we will be talking about in coming lectures. So DNA and RNA are nucleic acids. They're made up of nucleotide monomers. A nucleotide is a sugar group, a phosphate group, and a nitrogenous base. We've talked about nucleotides before. Go back to the organic molecules lecture and you'll see that. The sugar phosphate groups form a backbone. They're sort of the sides of the ladder. The nitrogenous bases are forming the rungs of a ladder. And the nitrogenous bases are all abbreviated with their first letter, T for thymine, C for cytosine, A for adenosine, and G for guanine. T and A always join together, and C and G always join together. So they have these binding patterns in the DNA molecule. This is kind of a little graphic here showing showing the, I guess this could kind of be a pointer. This is a pointer. Anyway, you can see the sides of the ladder here. If you can see the yellow thing moving up and down, we'll have to replay this to see if that works. But those are the sides of the ladder. They would be the sugar phosphate groups. And then the rungs of the ladder in between would be the bases, C with G or T with A. And then you give it a double twist, and that gives it the double helix look. These are three models of the DNA molecule: the ribbon model, the chemical structure, and the computer model. Of these, the computer model, is the space filling model. So it is more of a true representation of what the molecule looks like. However, you get a little bit more information from the chemical structure. It shows the binding patterns of C with G and T with A. T and A bind with 2 hydrogen bonds. C and G bind with 3 hydrogen bonds. And you can see that sugar phosphate groups are forming the sides of the ladder. The ribbon model also kind of shows you that. The ribbon model shows you a little bit better of the shape of the overall model. So there's 4 different nitrogenous bases. 2 of these are called pyrimidines. They are T and C, thymine and cytosine. They have a single ring structure. And 2 are called purines, A and G. They are a double ring structure. Adenosine and guanine. T always attaches to A with 2 hydrogen bonds. G always attaches to C with 3 hydrogen bonds. And that's for DNA. RNA is a little bit different because RNA uses no thymine. Instead, it uses U, which stands for uracil. So in RNA, you have uracil attaching to adenosine rather than thymine attaching to it. Here I was going to draw a little strand of DNA for you. So if you had the binding pattern, let me see, if our parental DNA strand parental, it's an A, parental DNA. So the parental DNA strand is going to be, G, C, T, G, C, T, T, A, G. So if we just make up that strand like that, the binding pattern of that would be your parental strand. Your new strand I'm going to put NS for new strand. G would match with what? G would match with, we can make the new strand a different color. G would match with C. G with C. T with A. G with C. G, A, T, T, and C. So that would be the binding pattern. The new strand is always going to have the corresponding base that matches with that parental strand. So if one strand is GCT, GCTT, AAG, the other strand would be CGA, CGAA, TTC. So that'll be something you'll be asked to do on the next exam. Okay, we'll probably have to insert a couple more of these slides in here into the PowerPoint in order to illustrate some stuff that I want to do that I would normally do on the board if we were meeting in person. Okay, the purines are going to be double rings. Adenosine has a double ring. Guanine has a double ring. The pyrimidines are thymine and cytosine. They have single ring structures. You don't have to memorize these structures. I just wanted to point that out. One double ring is always going to match with a single ring. That keeps the 2 sides of the ladder of the DNA molecule equidistant from each other. And that's important. That also helps to keep down errors in transcription, errors in the copying of DNA from one molecule to the next. Having that equidistance is important. DNA replication. DNA copies itself by first unzipping the 2 strands using an enzyme. The enzyme is called helicase. And this creates the 2 parental strands, which then serve as templates, attracting free nucleotides. So free nucleotides are in solution, and those nucleotides join kind of join in the party and make the new daughter strand. So each parental molecule will make each strand of the parental molecule will unzip and make two new strands of the new DNA. So this is called the semi-conservative model because each new DNA conserves one strand of the parental strand and makes 50% of it will be a new strand made out of these free nucleotides that are in solution. The process is very complex. We're not going to go into it in a whole lot of detail, of course, but it can be done very quickly. E. coli, for example, can replicate 4.6 million DNA base pairs in under an hour. Humans have about 6 billion pairs, and we require only a few hours to replicate our DNA completely. So it's a very quick process. And what makes this process quick is that it starts in multiple areas and replicates in both directions. So we'll get to that in just a minute. This is just showing the untwisting of a DNA molecule. The parental strand is shown in blue. The new forming strands are shown in gray. And so they will just attach as nucleotides. So that would be the G with its sugar and phosphate group would attach to the C, and the G with the C here, and the T with the A, and the G with the C. And then they bind together. Their sugar phosphate groups would later bind together. This is just another picture of it without showing the twist. So the parental model breaks in half, and then free nucleotides come and they join together, and G will always be matching with C, C with G, T with A, until you have two identical daughter strands, and these identical daughter strands will then possess 50% new DNA and 50% of the old parental DNA. So that is called semi-conservative replication. So this is what I was saying earlier. There's multiple origins of replication along a DNA strand, and these multiple origins of replication are often called bubbles because as the two strands start to unwind, they kind of form, they kind of look like a bubble if you were seeing them under a microscope. And then the DNA copies itself in both directions simultaneously. And this is very important because it really shortens the overall time needed for replication. If it just started at one end and then replicated all the way through, it would take a lot longer for that to occur. So this kind of shows this here. You've got the blue again is the parental molecule. Gray would be the new daughter molecule forming. And the gray molecule is going to form, it's starting in like 3 different areas here. They're showing 3 origins of replication. And then you see these little green arrows going in both directions. Those indicate that replication is taking place in both directions. So those are called forks, each direction that it's going. So it has 3 bubbles here and 6 forks. Anyway, in the long run, you're going to get 2 daughter DNA molecules. They're going to be semi-conservative, retaining 50% of the parental DNA and 50% of brand new DNA. And because it starts in these multiple places, remember the important thing about that is that it's going to shorten the time, the overall time for replication. Yeah, the important thing, sorry, I had a brain fart there. The important thing about the 3' end and the 5' end is that one strand is made continuously and the other strand is a lagging strand. And so that's what I want to show you, and that's what I want you to understand for the next test. So here we have the 3' end over here with the hydroxide group, which is attached to the sugar group. Notice that the sides of the ladder here, sugar, phosphate, sugar, phosphate. And then on the other strand that says the 5' end, it's saying phosphate, sugar, phosphate, sugar. So the 5' end is the phosphate group. The 3' end is the hydroxide group. And the new strand will always grow from the 3' to the 5' of the parental strand. So it's growing from its 5' to its 3', if that makes sense. So one strand is made continuously, and that's the 5' to the 3' strand, and that's going to be called the leading strand. The others form in little bits called Okazaki fragments, and these are called the lagging strands. The Okazaki fragments are later joined together with an enzyme called DNA ligase. So here, if we had the two parental strands, you can see the 3' to 5' end here at the top. And as the molecule is unzipping, then this 5' starts to grow. The new strand growing from the 3' of the parental strand, it's growing from its 5' to its 3'. So that one can be made continuously because it's growing in the right direction. It's growing from the 3' to the 5', so it can just grow continuously. Now the other strand is called the lagging strand, and the lagging strand has to be made in little pieces. These little pieces have to go in that direction because they have to form from the 3' to the 5' end. So they basically have to wait for the DNA molecule to unwind more. So they're going to be made in these little pieces, and then these pieces have to be joined together again later. And the joining together later is going to be by an enzyme called DNA ligase. And this green arrow at the bottom here shows the overall direction of replication. So as the molecule, as the blue molecule, that's the parental molecule, as it begins to unwind, one strand can be made continuously. The other strand has to be made in these little pieces. And that's because they have to have this binding pattern. They have to go from the 3' to the 5' end. And that's what makes for a leading strand and a lagging strand. I should probably draw something there. Okay, so that last part seemed kind of confusing. So let me try drawing this here. Hold on, let me shut the door. Okay, let me try drawing this for you because I think that last slide was really confusing. So here's our parental strand in red. Okay, so it's unzipping, unzip. And then the new strand is going to be growing. So this is, here we have the 3' end of the parent and the 5' end of the parent. So this new strand is going to be growing, sorry, in this direction. And that's going to be continuous. So that's called the leading strand. It's going to form continuously because it's forming in the right direction. Leading strand. Then the other strand, because this is down here, we did this in blue. So this will be the 5' end and the 3' end. So this strand, this new strand, is going to have to grow in little segments. So it has to wait for this to unzip, the parental strand to unzip further to make more strands. So this is called the lagging strand. Lagging strand. R, D, those are G's. Okay, so that's the important thing. For the test, you want to know that the leading strand is made continuously. The lagging strand has to be made in these little sections. And what these are called, each of these lagging strands is called an Okazaki fragment. And the Okazaki fragments will then be bound together later with a DNA ligase. DNA ligase. Which is another enzyme, of course. The ASC indicates an enzyme. So hopefully that made a little bit more sense than the last slide. So the leading strand made continuously, the lagging strand has to be made in little pieces, and that's called Okazaki fragments. I'm going to have a video that you guys are going to be able to watch too, and that should help a lot with this particular learning segment. Okay, now proofreading. Sometimes errors are made when replication occurs. There's polymerases, other enzymes that actually scan along the DNA molecule. And when they catch errors, they snip those out and put the right bases in place. So this is another way that the DNA replication is controlled. Errors are controlled in the DNA replication. Okay, so this is going to take us to protein synthesis, the flow of information from DNA to RNA to protein. First of all, I just want to mention that the reason that the body makes so many proteins is because we use proteins for everything. They're enzymes, they're immunoglobulins to provide defense against invaders like the coronavirus. Globins that carry oxygen into our cells and CO2 out of our cells. Transporters, they control osmosis, they control the those transport proteins that bring things in and out of our cell membranes that we talked about. Of course, structure, cartilage, nails, hair, muscles to control movement, hormones that cells communicate with other cells using hormones, and these can affect our blood glucose level or water retention. So basically, we're a big mass of proteins. We have to make lots and lots of proteins to do lots of different things. So let's talk about how those proteins are made. First of all, DNA inside the nucleus of the cells, DNA is transcribed into an RNA. This RNA molecule is then transformed. Little things are altered with it, and it's turned into an mRNA, a messenger RNA molecule, which will then leave the nucleus and go on to form the protein. And then the RNA is translated into the protein. It's called transcribed from DNA to RNA because you're transcribing from the language of nucleotides to the language of nucleotides. And RNA to the protein is called translation because you're translating the language of RNA, the language of nucleotides, into the language of proteins, which is a language of amino acids, because remember, amino acids are the building blocks of proteins. The actual presence of a particular protein determines the phenotype of an organism. So what's a phenotype? A phenotype is the outward appearance, whether I'm tall or short, whether I have brown hair or blonde hair. These are all determined directly from the proteins that are made. So ultimately, it's the DNA that regulates the proteins that are going to be made, but it's those actual proteins that directly determine the phenotype. And we'll talk more about phenotypes when we talk about genetics. Okay, I'm going to stop again there. Okay. So DNA, this purple area here is representing the nucleus, and you could see the DNA double helix up at the top. So first, DNA is transcribed into an RNA strand. And there's your RNA strand shown in pink. And that's called transcription. Write that down. You'll need to know that for the test. And then translation would be when that RNA strand, once it gets turned from a basic RNA strand into an mRNA, a messenger RNA strand, it will then leave the nucleus via one of those pores that's in the nuclear membrane that we talked about earlier in the class. It will leave that pore, and then it will go out and it will bind with a ribosome, which we're going to talk about in a minute, and that is where the protein will be made. So that protein that's showing at the bottom there, that little string of beads, of course, that would be the string of amino acids that's being formed. So transcription is basically the rewriting of the DNA code into RNA. It's called transcription because it's using the same nucleotide language, the language of nucleotides, nucleotide to nucleotide, DNA to RNA. Each word is a codon consisting of 3 nucleotides. Now, as I go through this PowerPoint, you're going to notice that I don't address all of what's written on the slide. You're welcome to read the slides without the voiceover. So I'm just doing this as I would in my lecture, and these slides are a little bit more wordy than what I usually go into. So bear with me on that, okay? Okay, so translation the switching of the nucleotide language to the amino acid language. So, of course, that's called translation because it's not the same language, nucleotides to amino acids. Each amino acid is specific for a codon. There's 64 possible codons. 61 of those codons are for amino acids, and 3 are for stops, just meaning to stop coding. The start codon is always AUG. I don't know why, but it is. Methionine is what AUG codes for, the amino acid that it codes for. Some amino acids have more than one possible codon. There are 20 different amino acids and 64 codons, so obviously you're going to have more than one codon per amino acid. This is a chart showing which codons are for which amino acids. Of course, you don't have to memorize this, but if I was to indicate here, I think I'm going to draw on this slide. Let's use purple for fun. Okay, so if you look up here, you've got the first base is on this code here. You have either U, C, A, or G. So whatever your first base is. So for instance, you would have CUU, that's going to be C. It brings you into this area here between these boxes. C is all this whole line. And then U would be the next one. This is going to be your second base, and this is going to be your third base. So if you had CUU, you would be in this block, and this block here. CUU is for leucine. So you can follow this and you can find any amino acid. So there's only 20 amino acids. So as you can see here on this chart, there's several that code for leucine: CUU, CUC, CUA, CUG. And this is important because this gives redundancy to the code. So redundancy I'm not going to fit it in here. Redundancy. Haha. Okay, so redundancy means that several codons can code for the same amino acid, but there's never any ambiguity, okay? CUU is always going to code for leucine. However, CUU, CUC, CUA, and CUG all code for leucine. And what that does is if there is a base change, a point mutation in that last letter, chances are you're still going to get leucine. You see that? And that's important. That also cuts down on the errors, that cuts down on the mutations that happen when DNA is processed into a protein. These are the table of 20 amino acids. Again, you don't have to memorize the amino acids. I did when I was in your place, and I don't remember them, so I wouldn't do that. But you can see that there's that common amine group. Well, the amine group is the NH3 group, and then you have this carboxyl group, and those together are common for every single amino acid. And then you'll see that there's a side group called an R group. It's called a side chain or an R group. I'm not really sure why it's called R, but it is. And that R group is what makes the amino acids different. So every amino acid shares that part that's shown in pink, and the part that's shown in blue is unique to every amino acid. So that means that some amino acids have different properties than other ones. They can, if you look down here on aspartic acid, it's polar acidic. If you look over here on lysine, it's polar basic. So some are polar, some are non-polar, some are slightly basic, some are slightly acidic, and this all gives them different properties. So when that string of amino acids, the primary structure of a protein, when it folds into that secondary structure of a protein, which is the alpha helixes and the beta sheets, and then that folds into the tertiary level of a protein, all those different binding togethers, the different hydrogen bonds that are going to form at the primary, from the, not the primary structure, but the secondary structure to the tertiary structure, Those are all held together by hydrogen bonds, and those hydrogen bonds are determined what binds with what by these different aspects of the R chain. Okay, so as we say, codons are redundant. And remember, a three-letter code is called a codon. 3 letters of the mRNA is called a codon, and each one codes for a particular amino acid. It's redundant, more than one codon for some amino acids, but it's unambiguous. Any codon for any amino acid only codes for that amino acid and not for another amino acid. The RNA strand, once it's perfectly formed, once it's finally formed is the word I was looking for, in the nucleus, once it leaves the nucleus, it's a fully formed mRNA strand, and it won't contain any spacers or punctuation. All the codons will be read in sequence. So here is a little cartoon of a DNA molecule. Each gene here is being shown. Each gene right here is being shown, 1 gene, 2 genes, 3 genes. Each gene is being shown in a different color. So one gene will then be transcribed into an RNA. So the DNA strand here is shown in blue. The RNA strand that would correspond to that is shown in pink. So again, remember there's no thymine in RNA. So A will bind with U. So AAA is going to be UUU. CCG is going to be GGC. GCA is going to be CGU, and then the 3 A's at the end are going to be 3 U's. And then those codons are going to be read in 3 base sets. So they're going to be read in 3 base sets like this, which it shows. And each one of these is going to code for a particular amino acid shown here. So if we look at it more closely, you can see that the DNA is transcribed into the mRNA, the messenger RNA. And then these are, I looked these up. UUU happens to be phenylalanine, GGC is glycine, CGU is arginine, and UUU is phenylalanine again. So that's what the particular amino acids are going to code for. And your primary structure will be formed, the string of amino acids. So the first step would be the transcription, the changing of the DNA into RNA. The 2 DNA strands, how that happens is 2 DNA strands separate. One strand is used for the pattern, and then it's going to code, depending on the DNA pattern, CGA, T, whatever it is going to code for the corresponding bases in RNA. And then, of course, it's catalyzed by an enzyme. And so the first stage is going to be initiation of RNA polymerase binds to a promoter, which tells the helix where to unwind and where to start transcription. As transcription continues in the RNA is formed from the DNA. That's called elongation. And then it's going to reach a stop terminator, and it's going to stop, and that's going to be the terminator. That's going to be where it stops the terminator sequence. And then it detaches from the template, and then a few things happen to it. It gets some things snipped out. It gets a tail and a cap put on it, and then it can leave the nucleus. We're going to see that in a second. So here's just kind of a little cartoon of it. The RNA polymerase here is the enzyme that starts it off. It joins with the promoter down here. And then as it does that, it's going to start making the RNA strand here according to the coding pattern. And then it's going to reach the terminator sequence. That's the terminator sequence. And then it's going to disengage and be a full-fledged mRNA. Well, a couple of things happen to it. It becomes an mRNA and leaves the nucleus. So this would probably be a good place to insert a slide, or we could just sort of do it over on the side here. If we had a DNA of, let's say, our DNA. Let's say our DNA is A, U, G, C, oh, shoot, it wouldn't have a U. Do. Hold on, let me get the eraser. A, T, G, C, T, A. Then your mRNA is going to be, let me see, then the mRNA. For A, you're going to have, U, because there's no T in RNA. For T, you're going to have A. For G, you're going to have C. C, you're going to have G. T, you're going to have A. And then for A, you're going to have U again. And then this is 2 codons. It's going to be read like this. 1 codon, 2 codons. So that's how it's going to be transcribed. And you can see here, if you look in closely at the RNA polymerase, you see the, this is the big enzyme that's on the outside here. And you can see that this is the DNA here in blue. The forming RNA is in pink, and it's formed by free nucleotides. These are nucleotides that are just in solution here, and they will match up depending on these patterns, A with U, U with A. The U is going to come in next, and U with A. And they're just going to join the party, basically, and make the RNA strand. So there's some processes that happen to the RNA strand before it becomes a full-fledged messenger RNA. I've been alluding to that a couple of times now. So the eukaryotic RNA has the interrupting sequences. The interrupting sequences are called introns. And they're separated by exons. So the introns do not code, the exons code. So first of all, a cap is added to one end. The 5' end gets a single guanine nucleotide. Don't know why, but it does. And then the tail gets this poly A tail. It can be 50 to 250 adenines long. So it's a long tail. The thought of why it gets this tail is probably due to the fact that the parts of that tail might get broken off, and so it gives it a little area to lose some adenines, to lose some of those nucleotides at the end, and not affect the sequence that needs to be copied. So you can have a little degradation on the front end or the back end, and I guess mostly the degradation happens more on the end where the poly A tail is, and that just helps to keep from the degradation happening and helps to keep the sequence from being fully copied. Helps to keep the sequence being fully copied. So then RNA splicing, you remove the introns, which are not being coded for, and the exons are then continuously joined, and that forms a messenger RNA. So if we look at that here, the DNA has these introns, and those codes are going to be snipped out, being shown here. They're being snipped out of the messenger RNA before it comes a messenger RNA. You get this single guanine cap, this adenine tail, and once it's all put together, then it's a full-fledged mRNA. mRNA. There it is. And it leaves the nucleus. Remember, the nucleus is the purple part. Outside the cell is the yellow part here. So it goes out through a pore, and that's where it's going to join with the ribosome. And that's going to be called translation. So translation involves a new RNA known as transfer RNA, tRNA. So tRNA actually has an attachment site for the amino acid, and it's going to come and bind with the mRNA, bringing that amino acid with it. And again, it has an anticodon that follows the codon sequence. So for instance, if your codon sequence is A, U, G, we all know that's methionine, and then we can have more A, C, let's just say. Okay, so if this is my messenger RNA, my tRNA, we're going to show you a picture of that in a minute. It's got like a little connection point. You have your amino acid up here. It's connected to it. It's got what's known as an anticodon. And this anticodon to read AUG would be U, A, C. And then it's going to read that first part of the codon, bringing its amino acid with it. Remember, C with G, A with U for RNA. So this is kind of what I was trying. It looks more like this in real life. That's a little hard to follow along to. It has an anticodon attachment down here. It has the amino acid site here, the anticodon down here. We're just going to use it in this cartoon form here to kind of follow along. So this is where the amino acids would attach, and that's going to be the anticodon down here. That's the anticodon. So translation occurs on the ribosomes. We've all learned that the ribosomes are the place of protein synthesis. A ribosome is a small organelle. It does not have, it's not bounded by a membrane. It has two components to it, a small and a large subunit. They're composed of ribosomal RNA, so that's another RNA. So you have the mRNA, which is the messenger RNA. Then you have the tRNA, which is the transfer RNA. And then you have the ribosomal RNA, which just makes up ribosomes. So messenger RNA, transfer RNA, ribosomal RNA. So these subunits are going to come together, and they're going to have a binding site for the mRNA and the tRNAs, and they're going to help to bring those things together in order for protein synthesis to take place. So the ribosomes are kind of a ball-like structure. This is sort of their protein structure here, what they look like, but you can't really see what's going on. So we'll make it into a cartoon, and now you can see that it has the large subunit here. That's in dark brown. The small subunit is in light brown. And the first step for the ribosome, before the ribosome even comes together, the small subunit will bind with the mRNA and when it binds, it's going to bind in such a place that the P site is going to be set up for AUG, for that methionine to come in. Doesn't want to make a G. Okay, there we go. And then it's going to have a new site available called the A site. And the next, when you have these codons down here, whatever they are, so we have C, C, A, U, C, G, whatever they are. So these are going to be in the next site. So this one here, 3 make a codon. And the tRNA that matches with that is going to come along next. I hope this isn't too confusing. Okay, so here you have the mRNA strand in pink. It's joined with the small subunit. The first tRNA, these are the tRNAs. The tRNA will come with the first amino acid. It joins in the P site. Then the amino acid will break off, join over onto the amino acid that comes in and joins into the A site. This amino acid will come and join with that one. This tRNA will leave, and this tRNA will move to the P site. So the one in the A site moves to the P site. Leave the A site available, and then it will read the next codon and bring in the next tRNA. So of course, it's in 3 steps. The initiation, where the mRNA binds to the small ribosomal subunit, and the first tRNA binds to to the mRNA at the start codon. The start codon reads AUG and codes for methionine, and then the first tRNA will then have AUC, which corresponds with that UAC, which corresponds with the AUG, right? Got it? Hopefully. And then the initiation, the large ribosomal subunit joins onto the party, so that you got the small unit and you get the mRNA, and then you have the large unit that joins and it's got the P site, and you have that first tRNA in there with the amino acid on it, and then you've got the second site here available for a new amino acid, which is the A site. So shown here, the small subunit mRNA attaching tRNA. The first tRNA comes in with MET, leaving the A unit open. And the next tRNA will read this codon and then bind at the A site. Then it's called elongation. Elongation will just be the making of the whole polypeptide, the amino acid, until it reaches a terminator sequence and then it disengages. And so you have the elongation is a codon recognition. That's where it's reading the part of the mRNA. Then the peptide bond formation, where that part breaks off. The amino acid breaks off from the one and joins to the amino acid on the one that's in the A site. And then translocation, where the one in the P site leaves, the one in the A site moves to the P site, leaving the A site ready for the next tRNA. So let's look at that in sort of a continuous form here. Again, you've got their mRNA, it's attached to the small subunit. The large subunit has come and joined in. You've got your growing polypeptide here in your P site. The A site's available. This anticodon will read this codon, and this amino acid will come in and join in the A site. The next thing that happens is this amino acid leaves from the P site, joins to the amino acid in the A site. Then this one, now it has no amino acid, it leaves. This one moves over to here, and it's got the polypeptide on it. And then that makes the A site available again for a new amino acid to come in. And it just keeps doing that over and over until the full protein is formed, and then it disengages. And that will be when it reaches a stop codon, as it's shown right here on the RNA. So, in review, does the translation represent DNA to RNA or RNA to protein? Translation is going to be changing of the language, right? So it's going to be this one, RNA to protein. Transcription is DNA to RNA. Where does the information per protein, producing a protein, originate? It's going to originate in the DNA. And then which one has a linear sequence? The mRNA. The tRNA is the one with anticodons that has the attachment site for the amino acid. The RNA just makes up ribosomes. And which directly influences the phenotype? Remember, we talked about proteins being directly responsible for the phenotype. So in review, we have transcription. The DNA is transcribed into a RNA strand. The mRNA has some interons snipped out. The exons are put together. There's a cap put on it, a tail put on it, and then it leaves the nucleus. It joins with the small ribosome down here, and the first tRNA comes and joins into the P site after the large subunit joins. It contains the amino acid. Then the next codon comes, joins into the A site. This amino acid gets attached to the one that's in the A site. This one leaves, and then this one that's in the A site moves to the P site, and then that's ready for another one to come along. So that just keeps going around and around. Here this shows the growing polypeptide being moved from this tRNA over to this tRNA. And then when it reaches the stop codon down here, The elongation is done. The polypeptide is now in its primary phase. And depending on what these amino acids are, it's going to turn into alpha helixes and beta sheets. And then it's going to turn into a three-dimensional shape. And those are all held together with hydrogen bonds. Okay. Mutations are any change in the nucleotide sequence. So remember that. Any change in nucleotide sequence equals a mutation. There are base mutations. That's replacement of one nucleotide with another. So changing a G to a C or a T to an A or G to a T, you know, anything like that is going to be a base substitution. A deletion or insertion is going to be removal of a base or adding of a base. So these are going to be less deleterious. That's going to be a long word. Less bad. Okay, less bad, because it's just replacing. You have like, okay, so you have A, C, or AG, C, T, G, G, C. So I can take that T out and I turn that T into a A. That's going to change the sequence. Now down here, a deletion or an insertion, so you start with your original A, G, C, what did I have there? T, I think, G, C, oh, is that a G2? I don't know. I can't read my own handwriting here. C, and then if you have a deletion, that's like boonk. Uh-oh. Now remember that everything's red in sets of 3. So now, since that one's missing, you have what's called a frame shift mutation. And a frame shift mutation means that the whole reading frame is going to be changed from that deletion or insertion on down. So here we had a G, we took the G out. It would have been AGC slash, that would have been red for one, but without the G there, it's ACT, and so everything's going to change on down the line. If you were to insert something, you're going to get the same thing. So we'll make an insertion, and now our reading frame is going to be AGA, CG, CTG, whatever base is at the end of this. So you see that it's a frame shift. So deletions or insertions are bad. More bad. More bad. Less bad and more bad. It's less deleterious and more deleterious, but obviously with my ability to write on this screen, we're going to just go with bad rather than deleterious. Mutations could be spontaneous, just due to errors in DNA replication. They can also be caused by mutagens, high energy radiation, chemicals, anything that causes like those little tiny gamma rays, little tiny wavelengths, just go into DNA and break the bonds. So that would be a high energy radiation. Chemicals can break bonds too. Anything that breaks a hydrogen bond can cause a mutation. Excuse me. So this is a base pair substitution. You start with, this actually is the basis of sickle cell disease. The normal hemoglobin, CTT, the mRNA would be GAA, corresponding to that. And so the normal hemoglobin would make leucine here. However, in the mutation, it's just 1 point mutation. You see there's just one change of a base here. T is changed to A, CAT, which changes this base, to GUA, which changes this amino acid to valine. Now, valine is hydrophilic, whereas glucine is hydrophobic. This changes the whole nature of the hemoglobin and is the basis for sickle cell disease. So sometimes a point mutation, just a small mutation like this, can cause a real problem. However, it often causes no problem at all because of the redundancy in the code. Remember, several different amino acids can code for the same several different codons can code for the same amino acid. So if the amino acid doesn't change, obviously the protein doesn't change. But if it does change, if the protein is different because of an amino acid change, if that amino acid has the same properties as the original amino acid, they'll probably be a functioning protein. It won't be a big deal. But if the amino acid that's substituted has very different properties, like is in this example here, hydrophobic to hydrophilic, then you can have a very deleterious effect. So generally mutations either have no effect or they have a bad effect. Every once in a while, though, a mutation can have a good effect and lead to an evolutionary novelty, which can actually lead to a good thing. So base substitutions are generally not as deleterious as base additions or deletions, which cause the result of a frame shift mutation, which I showed earlier. And a frame shift changes every amino acid sequence from that point on, resulting in a very different and usually non-functioning protein. Mutations are more serious when they occur in meiosis, leading to abnormal eggs or sperm, than in mitosis, or in a mitosis when it's a developing infant. But if you have like a defect in one of your skin cells or something, you might end up with a mole. It's not going to be a horrible thing. But every cell that comes from that cell with a mutation is going to carry that mutation. So if it's a deleterious mutation, if it's in a germline, or if it's in a developing infant, it's going to have a much bigger effect because more cells are going to be made from that cell. So this shows the difference between a base substitution here or a deletion. So as you can see, it's a frame shift. Met, lysine, phenylalanine, serine, alanine. So if you just changed a base here, it only changes one amino acid. If you stick a new one in or pull one out, this here is a deletion. So a substitution is going to change one amino acid. A deletion or addition is going to be a frame shift mutation. And that's going to change every single amino acid after that because it's going to be changing the way that it's read. So I hope that's clear. And as I said, most mutations are actually neutral. They don't cause any benefit or harm. But every once in a while, one's beneficial and leads to an evolutionary advance in an organism. So that's how evolution can occur, is with mutations.